So hello students, all of you, welcome back to your own channel Vedantu NEET English. Right, this is Basavaraj Sir, your biology master students. Welcome all of you. Now, this is a part 2 of your Biomolecules chapter. Right, in this chapter, in this second part, if you do not know what is carbohydrates, if you do not know, students, one second, let me just mute myself here. Okay. One second, students, one second. Yes. If you do not know what is carbohydrates, if you do not know what is a catalyst, what is an enzyme, how does the enzyme function, what is Vmax, what is half Vmax, and what is Km value, how do you think enzymes reaction work, what is the active site, what is activation energy, what is a cofactor, what is a apoenzyme, what is a holoenzyme, every single thing in this particular class we'll be learning, students. It doesn't matter if you do not know anything, doesn't matter, because I will be teaching you from the scratch. I'll make sure, students, I'll make sure at the end of this video, I'll make sure, students, at the end of video today, at the end of video, you will learn everything, students. You will learn everything. Okay. Yes. Hello, students. Hello. Students, if I'm audible and visible, students, if I'm properly audible and visible to all of you, quickly add some fire emoji in the chat to let us know that yes, class has been started, class has been going on right now. All of you students, quickly fire emoji, students, quickly fire emoji in the chat. Now, first things first, before we go to enzyme, we have a small thing there. Very small topic called as carbohydrates. Okay, something called as carbohydrates right now. Now, let's understand what is carbohydrate, students, first of all. Okay, students, let's understand what exactly is carbohydrates, right? Now, what if I say, students, carbohydrates are nothing but, they are nothing but hydrates of carbon, or I'll give you a better definition. Carbohydrates are your chemical compounds. They are the chemical compounds with carbon, oxygen, and hydrogen. So all of these elements are joined together to give us a chemical compound. Carbon, oxygen, and hydrogen are combined together to give us a chemical compound, and that chemical compound is called as carbohydrate, student. Carbohydrate. Now, all of you might be aware of a few carbohydrates, such as your anyone who knows what is a glucose? Anyone who knows what is a fructose? See, you might have heard somewhere in class 10th. Don't worry, I'll teach you class 10th also, right? Somewhere in class 10th, you might have heard glucose, fructose, sucrose. Yes or no, students? Yes or no? Have you heard about them? Now, all of these glucose, sucrose, fructose, everything goes, right? All of them are nothing but carbohydrates. Carbohydrates, students. Now, students, all of you, if you look at a structure of glucose, or if you look at the structure of your fructose, can all of you notice they have multiple OH bonds everywhere? Multiple OH, yes or no? So in a carbohydrate, we can find something called as multiple OH groups. Multiple OH groups can be seen here. Multiple OH groups can be seen here. Students, apart from that, students, they will also have something called as functional group called as aldehyde or ketone can be present. Now, if you know basic chemistry, basic organic chemistry, you can see there is CHO here. That is the aldehyde group here. And if you notice at your fructose, it has a ketone group here. Ketone group. So, let me just summarize everything right now. Carbohydrates are chemical compounds which has hydrogen, carbon, and oxygen. They have multiple OH groups everywhere, and they have ketone or a aldehyde group. That is basically your carbohydrate. Now, students, I want all of you to understand. Can anyone tell me, is these carbohydrates, are they present in filtrate or are they present in retentate? Anyone, students? Anyone? Where do you think carbohydrates are present? Do you think carbohydrates are present in the filtrate or retentate? Now, the actual answer is very tricky. Why? Because they're present in both. They are present in both. They are present in your filtrate also. They are present in the retentate also. So they are present in both actually. Now, how can they be present in both? Very simple answer, student. Very simple answer. The reason we find them in filtrate as well as retentate is because carbohydrates can have multiple forms. There is something called as simple carbohydrates such as your monosaccharides, disaccharides. Then we also have something called as complex complex carbohydrates that mainly includes your polysaccharides and oligosaccharides. So students, the next topic is the different types of carbohydrates. Okay students, see this. There are different types of carbohydrate. Can you see? There is monosaccharide, there is disaccharide, there is oligosaccharide, and there is polysaccharide. There is polysaccharide, students. Okay. So I want all of you tell me right now, have you heard about them? Tell me honestly, tell me students in the chat. Yashashwini, I'll teach you, don't worry, 10th, I'll teach you, don't worry. Right? I'll teach 10th also, I'll teach 9th also. Okay. Now, students, has any one of you heard about them? Mono, di, oligo, poly in your life somewhere in any textbook also? You might have heard. So let's go over them little bit, not very detailed. Overview about all of them, students. Overview. Okay. Now, students, here the first one is called as a monosaccharide. Okay. Now, students, all of you, what is the meaning of mono? All of you. Mono means 1, 2, 3, 4 or 10 or 7 like tala. Tell me students, what is the meaning of mono? Mono means 1. Yes or no? Mono means 1. So here, in the case of monosaccharides, how many types of monomers can you find, students, here? Anyone who can tell me? If it monosaccharide means 1, so technically speaking, we will find only a single unit here, a single monomeric unit, which cannot be divided further. Okay, so it is the simplest carbohydrate, very simple, forms the building block of large carbohydrates. That is, when you join monosaccharide, we get a polysaccharide. Okay, now cannot be hydrolyzed into smaller units because it is actually the smallest. It is already the smallest, so you cannot make it even more something smaller. Not possible, students. Okay, now here are the few examples. Here are the few examples of your monosaccharides. Okay, monosaccharides will have a single sugar unit. Okay, you can have 2 carbon, 3 carbon, 4 carbon, 5 carbon, right? 6 carbon. So carbon number doesn't define the sugar unit. This entire thing is one sugar unit. So can you see, students, here it has one aldehyde group here, so that is called as monosaccharide. Now students, now what do you think disaccharide is? Di means 1, 2, 3. What is meaning of di? Di means 2. So disaccharide, saccharide is basically sugar unit. Di means 2. And saccharide basically means sugar. Is written here. Okay, so in the case of disaccharides, we can find 2 sugar units. Let's say I have 1 sugar unit here, that is your 1 monosaccharide. I have one more monosaccharide here, that is 2. This is first sugar unit, this is second sugar unit. Now, how do we join them, students? How do we join them? This sugar unit and this sugar unit, they are joined together by a bond here, students. What is the bond? Friendship bond? No. What is love bond? No. What is the bond, students, here? The bond is called as glycosidic bond here. Okay, so they are one monosaccharide, second monosaccharide. So disaccharides here. These disaccharides are bound together by glycosidic bond, all of you. Okay, glycosidic bond. Now in examination, students, what will they ask you? In examination, they will ask you the examples of disaccharides and what are they made up of? Like, what are they actually made up of? Now, for example, all of you, first one is called as sucrose, second is called as lactose. All of you, have you heard about them, students? Have you heard about them? Sucrose and lactose? Yes or no? Now, students, here, what is sucrose made up of? Sucrose is actually made up of 2 units. Obviously, it is a disaccharide. If sucrose is a disaccharide, it has to be made of 2 units. Unit 1 is called as glucose. Unit 2 is called as fructose. So, 2 units, glucose and fructose, they are joined together by glycosidic bond. Okay, glycosidic bond. Now, students, can anyone tell me, anyone in the chat tell me, is glucose and fructose monosaccharides or disaccharides? Individually. Use your brain. All of you use your brain, students. All of you use your brain. Tell me, is glucose and fructose individually, are they monosaccharide or are they disaccharide? They are monosaccharides. So individual monosaccharide, join them together, tada, disaccharide. That is very simple. Now similarly, lactose, so you can see lactose. In lactose, glucose is again one monosaccharide. One more, galactose is one more monosaccharide. So one unit, second unit, they are joined together by glycosidic bond, and that will give us lactose. Okay, students, that will give us the lactose. So students, can anyone tell me two examples of disaccharides in the chat quickly, students? Two examples of disaccharides quickly, students, in the chat. Two examples of disaccharides. Anyone? What are the two examples of disaccharides? All of you. One is sucrose, other one is lactose, right? So sucrose and lactose are the examples of disaccharides. Perfect, perfect. Then there is oligo, students. Oligosaccharide. Now in the case of oligosaccharide, it can have around 3 to 9 monomeric units. That is 3 to 9 sugar units. Like monosaccharide. All of them are joined by glycosidic bond. Very simple, students. So example is raffinose. This example you can remember, that's all. Raffinose is actually 3 monomers. That is, raffinose is made up of glucose plus galactose plus fructose. 3 monosaccharides joined together, that will give you raffinose. Okay. So all of you can remember one example of oligo. All of you can write. All of you in the chat would answer. Raffinose, all of you can remember. Raffinose has 3 monosaccharides. One is called as glucose, galactose and fructose. 3 different monosaccharides joined together in the case of your raffinose. Now students, now what is the most important, student? The most important is called as your polysaccharide, students. Polysaccharide. Now students, now please understand polysaccharide. Now students, what is the meaning of poly? Anyone? What is the meaning of poly? Poly means what? Many. Poly means what? Poly means many. Technically speaking, it is more than 10. It is 10 or more. 10 or more. Because anything below 10 is oligosaccharide. Anything below 10 is oligosaccharide. So 10 or more monomeric units, that becomes a polysaccharide. That becomes a polysaccharide. That becomes a polysaccharide. Okay. It is, students, basically a polymer of monosaccharides. It is a polymer of monosaccharides. Long chain. For example, you can see here, students. Let's say monosaccharide. So basically anything more than 9. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 we'll see. Okay. Now this right now is a polysaccharide. Student. Polysaccharide. Now students, please remember, I'll teach you also in the next few minutes. It has something called as a reducing end and non-reducing end. I'll teach you all this. What is reducing end, what is non-reducing end. So please remember this much. Okay. Now students, now all of you in your school days, did you have something called as groups, houses? All of you. See, I was in almost all the houses. Like in my 5th standard, I was in your blue house. No, 6th standard, I was in blue house. 7th and 8th, I was in your yellow house. 9th and 10th, I was in your green house. 11th and 12th, I was in red house. Okay. So I was almost in all the houses. Okay. Students, which house you were in, all of you? Sharmita, Adin, UV, Rao, all of you. Which house you were in, students? Which house? All of you. Do you have that houses or not? First of all of you, do you have the house? Red house, green house, blue house. See, I was in everything. So throughout the year, because they used to shuffle in our school. In our school, they used to shuffle. Right? All of you. Yellow house, huh? Huh? Which house, students? Which house? There used to be color coding. No. Okay. Now, students, usually what happens in school assembly? In school assembly, generally speaking, you stand according to classes. Yes or no? This is, let's say, 9th standard, 10th standard, 6th standard, 7th standard, 8th standard. Now, when you're standing class wise, in that class, all the different houses are in one line only that time. Yes or no, students? Yes or no? When you stand, let's say your classes, you are in 12th standard now, 12th standard. When you're standing on long line of 12th standard, all of you belong to different houses. But in normal days, you're all standing together. But on the special color day, you can stand according to houses, right? All the red house, one side. All the green house, one side. All the blue house, one side. Now, what do we understand from this? What do we understand from this? See, when everyone is standing according to the houses, every single one of you are same color, students here, same color. Now, in the case of your polysaccharide also, in the case of your polysaccharide, if all of these, let's say this is a polysaccharide, this polysaccharide is made up of monomers. If all of these monomers is only one, like you have red color, let's say, student, let's say all of these monomers are basically, let's say, glucose. Now, such a condition, student, such a condition is called as homopolysaccharides. Homopolysaccharides, students. In the case of homopolysaccharide, we have only one type of monomer. Only one type of monomer. Only one type of monomer. Now, students, all of you, this you can tell. So, what is this now? Students, in the case of heteropolysaccharides, in the case of heteropolysaccharide, can I tell we will have different types of monomer joined together? Let's say here I have glucose, yes or no? Let's say here now we'll have fructose then we have again glucose, then we can have again, let's say, some other monomer. Let's say we have maltose here, then again a glucose here, students. Yes or no? Let's say here we have again a fructose here. Let's say we have tetrose. So basically, in the case of heteropolysaccharide, there are different monomers. Done. Now, students, here is an example, students. If you look at the peptide bond, if you look at your protein also, protein is made up of different types of amino acids. It is a heteropolymer. It is a heteropolymer itself. Now, students, please understand. One second, students. Okay. Now, so all of you need to understand. Let's take polysaccharides. Students, one second. Students, one second. Okay. Maltose is disaccharide. I used by mistake the maltose there. Okay. Now, students, let's take all the polysaccharides. If you can divide all the polysaccharides into two categories. Okay. Let's say we have homopolysaccharide as well as we have heteropolysaccharides. Now, all of these homopolysaccharides right here, all of these homopolysaccharides can be divided into two categories mainly. Okay, all the homopolysaccharides can be divided into two categories mainly. Okay. We need a bigger space here. Okay. Now, students, once again, let's just go back and erase this maltose here. Let's say here also we have a fructose. Okay. Now students, we have two categories here. Okay. Now one is based on structure. Right. Based on the structure we have, then we based on storage we have. Okay. Now students, see this. Based on structure, we have two types. One is called as cellulose. One more is called as chitin. Cellulose and chitin. Two types under structure right now. Now, under storage, we have three types actually. One is called as your starch. Second is called as your inulin. And third one is called as glycogen, students. Glycogen. So under homopolysaccharides, from structure we have two types, storage we have three types actually. Okay. Now under now students, under your heteropolysaccharide, very simple students, very simple. There is no much classification here. I'll give you two different examples here, students. I'll give you two different examples. Okay. Here the first one is called as your peptidoglycan. Students, all of you tell me, you might have learned, right? In the case of bacterial cell, students, in the case of bacterial cell, you might have learned peptidoglycan is made up of what? Peptidoglycan is the outer layer of, it's a layer in your bacteria, remember? Right? The cell wall is made up of peptidoglycan. Peptidoglycan is made up of something called as NAG and NAM. It is made up of what? Something called as NAG and NAM. Okay, N-acetyl muramic acid, N-acetyl galactosamine, right? Glucosamine, right? Glucosamine, right? Two different types of your sugar units. So when there are two different types of sugar units, it is a heteropolysaccharide. It is a heteropolysaccharide. One more example is agar-agar actually. Agar-agar is one more example. Okay. Now in examination, what they ask students? In examination, they can ask you directly this only. In examination, they can ask you directly this. What are the functions? They can ask you this entire, let's say they ask you chitin. Students, is chitin a heteropolysaccharide or homopolysaccharide? Is chitin a heteropolysaccharide or a homopolysaccharide? All of you in the chat. What is chitin? Chitin is a heteropolysaccharide or homopolysaccharide? You will tell me, sir. Chitin is a homopolysaccharide. Very simple from here, you can tell now. Okay. Now students, here are all the functions given to all of you. Functions given to all of you students. Can you see? Starch, it is a polymer of glucose. So starch is made up of long chain of glucose. Starch. Same goes for glycogen. So starch and glycogen, students, they are long chain of glucose. Long chain of glucose here. Long chain of glucose. Okay. There. Yes. We'll make calculations. We'll make calculation session. We will do numerical session. So starch and glycogen both are made up of glucose. Long chain of glucose. But inulin is special. In this was a PYQ to all of you. NEET PYQ. What is inulin a polymer of? Inulin is a polymer of, students, fructose. Can you see here? Inulin is a long polymer of fructose. Long polymer of fructose. Okay. So you can write here also. Let's write here also. Long polymer of fructose right now. Fructose right here. Okay, fructose. That is the different storage units. Different types of your storage units. Now students, inulin is generally found in the case of your plants, right? Starch. Students, where do you find starch? In the case of plants or animals? Where do you find starch? Starch is generally found in the case of your plants again. Okay. Then we have glycogen. Glycogen is a storage in the case of animals. Now students, please remember, this starch and this glycogen almost have a similar structure. The only difference is that glycogen will have more alpha 1,6 branching. The alpha 1,6 branching is very often in the case of glycogen. So glycogen will have more complex structure compared to starch. Students. All of you should know there is starch has something called as your amylopectin and amylose. Amylopectin and amylose are two parts of starch. Okay. You can see here, right? Starch, not metabolized in human body, stored in the helia, basically in the case of plants here. Now cellulose, students, cellulose is structural type. It is a polymer of glucose again. Polymer of glucose only. This is polymer of glucose, right? Now chitin is a polymer of N-acetyl glucosamine. So chitin is a polymer of. So they can ask in examination. Students, what is chitin polymer of? So please remember, chitin is a polymer of your N-acetyl glucosamine. That is chitin made up of. And where is chitin found? Chitin is found in the exoskeleton of your arthropods, that is insects. So exoskeleton is made up of your chitin. Okay, so that is your structural type of your homopolysaccharide. Students, homopolysaccharide. Now students, this is your heteropolysaccharide. Can you see this right here is heteropolysaccharide. Here we can see NAG and NAM. These are NAM, these are NAG. So NAG and NAM can be found in the case of your peptidoglycan. The same goes for agar-agar, students. Agar-agar is made up of galactose and 3,6 anhydrous L galactopyranose. This they'll not ask in examination, but just please remember, agar-agar is a type of your heteropolysaccharide, students. Okay. Now, can anyone tell me, students, where do we get agar-agar from? What is the source of agar-agar, students? What do you think, Vignesh, Sharmita, Abhinya, tell me, what is the source of agar-agar, students? What is the actual source of agar-agar? Now, all of you know, if you have studied a chapter called as Plant Kingdom, there under algae, when we study about rhodophyceae, that is red algae, there is Gracilaria and Gelidium. Gracilaria and Gelidium will provide this agar-agar. And this, students, this agar-agar is used in microbiology. It is used to make ice creams. It is used to make jellies and everything. That is agar-agar. Okay. Now, students, with that being done, shall we do a small experiment, students? One small experiment is there. A very, very small experiment. All of you should know what is a reducing sugar. What is a reducing sugar, students? Very basic thing is there. Please remember, all of these reducing sugars, they will have a free aldehyde or a ketone group. That is called as reducing sugar, students. See, they have a free aldehyde or a ketone group, which can undergo reaction generally. That is called as reducing sugars. You might have learned this in chemistry also, no? Students, have you learned it? Did Diksha ma'am teach you all the students? Has Diksha ma'am taught you? Or should I tell Diksha ma'am that you don't know? All of you? Yes? Diksha ma'am taught, right? Diksha ma'am has taught, I feel. Ma'am would have thought. Okay. So reducing sugar will have free aldehyde or a ketone group. Reducing sugar reduce the cupric ion to cuprous ion. Very simple experiment. Very simple experiment is there. You have to take 2 ml of, in the test tube, add 2 ml of your Benedict solution. 2 ml of Benedict solution and 2 ml of, let's say we add a type of your monosaccharide. Okay. 2 ml of your monosaccharide, let's say glucose. Let's say we add 2 ml of glucose into this and 2 ml of Benedict solution here. Now, after some time, the color of the Benedict solution will become brick red. Why brick red, students? Because all the cupric ions, right? All the cupric ions in this Benedict solution will become cuprous because of the free aldehyde and the ketone group. So that is a reducing sugar. Non-reducing sugar will not have this free aldehyde and ketone group. Okay. Now, students, if you see your all the polysaccharides as well as your couple of your disaccharides like sucrose. Okay. Now here they will have a reducing end and a non-reducing end, students. Now because of this non-reducing end, they will not give this brick red color. They will not give you the brick red color here. So these are your non-reducing sugars. All of these are your non-reducing sugars, polysaccharides as well as your disaccharides like sucrose. Okay. Now students, there is one more experiment. Okay. Now I'll teach you very simply. Okay. Now let's say I have a cricket ball. Okay. Now I will throw the ball at you. What will you do, students? Can you catch the ball? All of you. Your hands are free. Can you catch the ball? All of you. If I throw a cricket ball at you, can you catch the ball? All of you, tell me. All of you. Hmm? If I throw a cricket ball at you, will you be able to catch it? Cricket ball. You're like, yes, obviously you can catch. With some effort, with some trial and error, you can catch someday. Okay. Now, students, what if I tie your hands? Students, what if I tie your hands? Like, what if I tie your hands like this? Like, I'll tie your hands and I'll throw the ball at you now. Now, can you catch? Now, can you catch? Don't tell me you'll catch with your teeth. You cannot. I will I will put a mask on your face. There's a mask on your face. So you can't even catch with your mouth also. Now, if I tie your hands, can you catch the ball now, students? All of you? You can't catch the ball now, obviously. Now, please remember that concept and let's apply it here. Okay. Now, all of you know there is something called iodine solution. It became very famous, no? Because of the paneer test and fake paneer. Okay. So let's take iodine, and to this one test tube, I will add starch. I will add here starch. Now, same iodine solution here. Solution here. Now, in this test tube, I will add cellulose. Students. I will add cellulose here. Right. So here I am adding starch. Here I am adding cellulose. Now, what happens? Students? Now, the first condition where all your hands were free, that is your starch condition. In the case of starch, the structure of a starch has something called as free helicals. Hands are free. So if hands are free here, can starch hold the iodine? Should all of it tell me students in the chat. If it has hands, if it has helicals here, can this helical hold the iodine? All of you? Yes or no? Like your hands are free, you can hold the ball, you can catch the ball easily, right? Now that is the case of your starch. So starch can hold I2 molecule in the helical portion. So positive test here. Or in this helicals, it can be held. Now students, now. Please understand students, please understand. What about in the case of cellulose? Cellulose is the condition where hands are tied nicely. When I have tied your hands. Now when I have tied your hands, can you hold the iodine? No. So this cellulose doesn't have any free helicals. So that is why it cannot hold I2. It cannot hold I2 molecule. It cannot hold iodine. Okay, students. So what is the answer, students? What is the answer for this question? This right here is a NEET PYQ, student. This right here is a NEET PYQ. Hello, Joshua. Welcome to the class. There's no DNA class now. Okay, students. Tell me, students. Cellulose, obviously the hands are tied. There is no helicals. Does not form blue color with iodine. It does not form blue color. Why, students? All of you. Tell me, students. Tell me why. What is the answer? It breaks down the iodine when it reacts with. It is disaccharide. It has helical molecule. It does not contain complex helicals and hence cannot hold iodine molecule. Obviously, students, option number D. Students. And students, please remember your chitin. All of those are called as complex polysaccharides. They're called as chitin is a complex polysaccharide. Okay. Now, students, here is a question. Let's see who can solve. Here are the different bonds. I have told students protein has what? Protein has peptide bond. Okay. Unsaturated fatty acids, they will have double bond. Saturated will have single bond. Okay. Nucleic acid will have, students, phosphodiester bond. Polysaccharides, we just now learned. Polysaccharides will have what? Glycosidic bond. No, glycosidic bond. So what is the answer? Let's see who tells first. So what is the answer, students? What is the answer? Students, what is the answer? Tell no. Don't keep it a secret. Do not keep it a secret. Tell me the answer. Done. Next question. It's always a NEET. All of these are NEET PYQs. Yes, I saw who told first. Tell me, students, tell me. Inulin is a polymer of fructose. Where is it, student? Where is it found? It is found in the case of your plants. Exoskeleton of arthropods is composed of. exoskeleton of arthropods is composed. So all of these are NEET PYQs. All of these are NEET PYQs. It's very easy for all of you to answer. Students, quickly smash your like button. The target is 50 likes. Or is it done? I don't know. Students, the target is 50 likes. Is it done already? It is not done, no? SIAD. Okay. So it is made up of, students, please be very careful. Please be very careful. Exoskeleton of arthropod. Okay. Now what happens is that when you're reading it, when you're trying to read it right, you will read this as chitin. It is cutin, something cutie it is here. So cutin is present here. So please be clear, it is cutin. This is chitin. So go for option number 4. So when you're in a hurry, right? When you hurry, right? Please don't go for cutin or chitin. Just go for chitin. Okay. Okay, students. Now with that, we come to the end of proteins. Now, students, here is a table here. This table is very important. The table is very important. This is a table of different functions of a protein. Now here especially GLUT4 has been asked a lot of times. Okay, GLUT4 has been asked a lot of times. Okay, GLUT4. That is, it enables the glucose transport into the cell. So GLUT4 will allow the glucose to enter inside the cell, students. Inside cell. Okay. Is it, students? DNA batch got delayed. Okay, let me just check. I only don't know. I think because some chapter was not over. So to make the chapter alignment, they would have made the changes, students. Okay. Nice. Now, students, see this. Glycogen, all of you know, intercellular ground substance in the body. Trypsin is an enzyme. Inulin is a hormone. Antibodies, like infective agents. Students, do you realize? Students, do all of you realize? Antibodies are proteins. Collagen, protein. Trypsin, protein. Insulin is a protein. So a protein can be enzyme, hormone, antibodies. All of these, students, what? All of these are proteins only. So do you realize how important protein is for our body, students? All of you realize how important protein is for our body? Yes or no? So if you need proper hormonal balance, we need protein. If you want proper hair growth, you need protein. If you need a clear skin, you need protein in your body, right? If you want to be healthy, we need proteins in our body. If you want a lot of enzymes to work, we need proteins in our body. So that is the importance of protein in our body. So unfortunately in India, our protein intake is very weak. Okay, students. So how many of you eat chicken, students? How many of you eat chicken? Students, how many of you eat chicken? Yes, students, how many of you eat chicken? Chicken, if you eat, protein. If you don't eat chicken, please eat a lot of paneer. Don't eat restaurant paneer. Obviously, buy the packet paneer. Okay, paneer, milk, right? All these are very good source of protein for all of you. Paneer, soya chunks. So eat a lot of protein in your life. At least every single day. Let's say your body weight is 60 kgs. If you are 60 kgs, at least 30 grams of protein you should take. At least 30 grams of protein you should take. 30 grams of protein. 30 grams of protein. If you're vegetarian, please eat a lot of dal, chana, chole, all that. Very important. So you need minimum to minimum, right? Half of your body weight protein grams you need. Technically, it's supposed to be equal, but very difficult to get it. Very difficult to get it. Okay, students, do that. Okay. So DNA batch updated CT1 syllabus. Update the CT1 syllabus. I'll check, students. I don't even what is CT1. Oh, I'll check, students. I'll check. Okay. Now, students. Now, all of you know this, students. All of you know this. Polysaccharides is done. This is your why iodine can't hold. Why your cellulose can't hold iodine. Okay. Now, students. Now we come to something called as structure of protein. Shall we start, students? Shall we start? All of you? Yes, I'll share that, students. I'll share the fish also very important. Fish is very important. Now, students, we'll understand something called as structure of protein. I'll get that. I'll send the notes. Now we'll study the structure of protein, students. Now, students, how many of you love chemistry, students? How many of you love chemistry? See, chemistry, they will see, if I ask a chemistry person, what is structure? Generally, they give me a 2-dimensional structure. Generally. Okay. Sometimes 3-dimensional. Physics people, they will also give you 3-dimensional only. But in students, in biology. But students, in the case of biology, what happens? You know that in the case of biology, we can see a structure. we can see a structure at 4 different levels. Crazy, biology students? Biology is crazy, no? So in the case of biology, we can see a structure at 4 different levels. 4 different levels. Now, as a matter of fact, students, as a matter of fact, protein has 4 different types of structure. 4 different types of structure, students. Now, all students, all of you understand what is primary structure, what is secondary structure, what is tertiary, what is quaternary, students. Please understand, students. Now, primary structure of protein is linear, long chain, just long linear chain, one dimensional, long chain, linear chain. That is primary structure of protein. Now, what do we understand from your students? What do we understand from your primary structure? From primary structure, we can understand something called as position of amino acids. We can understand position of amino acids. All of you, we can understand the position of amino acid. Like for example, here we have leucine, isoleucine, third one. Let's say we have aspartame here. We have here different methionine here. So we can find basically, students, with the help of primary structure, we can understand the position of amino acid at what do we have at 1, what do we have at 2, 3, 4, 5, 6, 7. So we can understand here. Okay. Now that is primary structure. Okay. Now students, in secondary structure what happens? Students understand. Okay. In secondary structure what happens? Secondary structure. Okay. Now students, in secondary structure what happens is that, students, these are my written notes always. Okay. In secondary structure what happens is that the protein chain is, this protein chain is there, no? Protein chain. It forms a plate here. It forms a plate-like structure. Now this plate-like structure can undergo something called as alpha helix in a right hand fashion like this, students. Can you see? It will start to rotate now. Now this is also the protein structure. It undergoes coiling right now. It undergoes coiling in a right handed fashion. That is alpha helix right now. Can you see? It undergoes coiling. Now this entire coil is made up of what? This, can you see this linear chain? The same linear chain is undergoing coiling right now. Coiling. Now that is called as alpha helix. Now what is beta plated, students? Beta plated and alpha helix both are part of secondary. Now in beta plated what happens, students? Let's see this. In beta plated what happens? Students, can all of you see this? Students, is it visible to all of you? This right now is called as what? This right now is called as beta plated structure of protein. That is called as what? Beta plated structure of protein, students. Beta plated structure of protein. That is, both are part of secondary, students. Both are part of secondary right now. Okay. Both are part of your secondary. Secondary now, students. Secondary. Now. What is tertiary, students? What is tertiary? Tertiary is basically making one ball. You have to make one ball right now. You make one ball. Very simple. You take this alpha helix, you take this beta plated, you take both of them and you completely make them a ball. Like a ball you make. Like a 3-dimensional structure you make. Okay. So you take alpha helix, take beta plated and make a ball. Now that becomes what? That becomes the tertiary structure of protein. And students, in tertiary structure we can find hydrogen bonding can be found here also. Here we can find hydrogen bonding here also, students. Apart from hydrogen bonding, we can find disulfide bond also here in tertiary, students. Okay. Now, what is quaternary then? If this is tertiary, what is quaternary then? Understand. Okay. I need students. I need some pages. One second. Let me just teach you with the help of some pages. I need some pages, students. Let me just take a book and we will do some sacrifice for science right now. We will do some sacrifice for science. Okay. Now students, understand. Let's say this right now is, students, understand. This woolen ball right now, this is tertiary structure. This is the tertiary structure right now. Now similar to this, students, similar to this tertiary structure, there can be more of them. Yes or no, students? Yes or no? Right? See, there can be one more tertiary structure. Okay. Now let's take one more now. Okay. See one more. Okay. One more is there. Now, if there are multiple tertiary structures, that is, this alpha helix and this beta plated, when they join together, they will give you tertiary structure, 3D structure. Now, this is third dimensional, right? Now, if you join all of them, students, when there are multiple tertiary coming together, this becomes a subunit, this becomes, second one becomes subunit, third one becomes a subunit. When there are three different subunits like this and you join them together, this entire thing is called as what? This entire thing is called as quaternary structure. That is called as what? Quaternary structure, students. Is that clear to all of you? What is quaternary? Now I'll give you more input, students. See this. This right here is your primary structure. Linear chain of amino acids. Linear chain of amino acids represents the position of amino acid. Which amino acid is present where? Here we have N terminal on the right side, C terminal on the left side here. N terminal, why? N terminal, students. N terminal is because there is amino group here. C terminal because carboxylic acid group here. So N terminal and C terminal. Now, secondary structure has your alpha helix, beta plated. This is alpha helix which has a right-handed fashion. Beta plated sheets of protein here. Okay. Then this is your tertiary structure, three-dimensional structure. Protein is folded upon itself. Protein is undergoing folding right now. And here all of the students, all the enzymes you studied are enzymes. Enzyme is a protein. But enzyme is technically which type of protein? Technically speaking, students, enzymes are your tertiary proteins. All the enzymes, all of you know, right? Enzymes. Enzymes are basically what? They're basically proteins only. Proteins. Now, all of these enzymes have which type of protein? They have a tertiary structure. They have a tertiary structure generally. They have generally have a tertiary structure right now. Tertiary structure. This is how tertiary structure looks like, students. Now, last is you have disulfide bond. Last is called as quaternary, students. Last is called as quaternary. The last is called as what? I'll teach students. I'll teach called as thread-like structure. Thread-like structure is this one. This is thread-like structure. Technically, this is thread-like structure. This is thread-like structure. Linear. If students try to imagine this individual thread here is a linear molecule. We take multiple linear and we join together. That becomes tertiary. Okay. Now here is a quaternary structure, students. Two or more polypeptide chains join together. Each individual one is a subunit right now. And arrangement of each folded polypeptide chain with respect to each other. Students, see this. I'll upload the notes, don't worry. One of the examples of tertiary is hemoglobin. Students, can you see the board? Students, this right here is your hemoglobin like this. This is how hemoglobin looks like. This right how hemoglobin looks like, hemoglobin. Can you see, students, here? This is one ball right now. This is one ball here. This is one ball here. This is one ball here. Where there are multiple subunits. Multiple subunits are joined together. Now this right here is called as the quaternary structure. This right here is called as quaternary structure. Okay, quaternary structure right now, students. Quaternary structure. Now, students, in this quaternary structure, if you see here, we have a quaternary structure generally has, in this case, hemoglobin. Hemoglobin, students, has two alpha chains and two beta chains here. Okay, so here we have the two alpha chains and we have two beta chains. They are coming together to give us a three-dimensional structure and quaternary structure here, students. Okay, so the example is your hemoglobin. Quaternary structure example is hemoglobin. Clear, all of you, students? Very easy to understand, no? Very easy to understand. Okay, it is made up of 4 polypeptide chains, totally 4. There are 2 alpha chains, 2 beta chains. Okay, then, students, all of you understood? Yes or no? Can I get a thumbs up? Students, can I get a thumbs up in the chat? So there are 2 alpha chains and 2 beta chains in the case of your quaternary structure. Now, students, 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 let me teach you. Students, let me show you my favorite protein. Students, let me show you my favorite protein. Students, which is your favorite protein, students? Which one is your favorite protein? My favorite protein right here is called as picachurin. This is a protein which is present in retina. It helps in transmission. It is actual thing. It is called as picachurin. It is a protein. Okay. Picachurin is a type of protein. It is my favorite protein because it sounds like Pikachu, obviously. Pikachu doesn't like. Right. Right, students. So it's a picachurin is a type of your protein here. Okay. Now, students, Students, you can read NCERT. It is very simple right now. It is very, very simple. Okay. Primary protein is what? Straight chain. Straight chain is primary protein. Then we have your secondary protein here. Secondary protein will have your alpha helix, where right hand fashion can be there. Other one is your beta plated sheets. Beta plated sheets are there. Okay. Students, then we have your tertiary structure of protein. Here what happens, students? Long protein chains is also folded upon itself like a hollow woolen ball, giving a tertiary structure, students. It is three-dimensional and biologically active for many proteins. Biologically active for many protein, students. Okay. Now, quaternary is multiple of them, students. Multiple of them. Okay. Very simple. It is very, very simple. Okay. Very, very simple, students. Now, one question arises, students. One very serious question is there on the board. Who can tell, students? Who can tell? All of you. Which of the following is most abundant protein in the animal kingdom? The most abundant protein in the entire animal kingdom is called as your... Students, anyone? Which is the most abundant protein? I'm not sure. Which is the most abundant protein, students? Anyone? Which is the most abundant protein in the entire animal kingdom? Animal kingdom, not biosphere. In the entire animal kingdom, we have the collagen. We have the collagen here, animal kingdom. Now, what if I say entire biosphere? If I say entire biosphere, that is the entire earth, then students, we have the rubisco. Rubisco in the case of biosphere, collagen in the case of animal kingdom. In the case of animal kingdom. Done? Done, students, done. Now, students, now there are different types of protein which all of you learned. Protein can help in defense, antibody. They can present as nutrient. They can present as hormone. GLUT4 for transport. But students, in all these different types of protein, right, there is something called as catalytic protein. A protein can help in catalyst. A protein can act as a catalyst. Now, what do you call that, students? If a protein can act as a catalyst, that is called as an enzyme. Okay, students, now we come to the next part of the chapter, that is called as your enzyme part. Shall we start? All of you, shall we start, students? Shall we start? Okay. Now, students, please understand what is a catalyst first of all, students. What exactly is a catalyst? Okay. Catalyst is a substance. Students, a catalyst will increase the rate of reaction or decrease? What will it do? Students, it is a substance which can alter the rate of reaction. It can alter, so it can basically increase or decrease. Both possibilities are there. It can increase or decrease. That is the main function of a catalyst. Increase or decrease, both possibilities, students, here. Now, catalyst can be of broadly two types here. One is called as inorganic in origin, that is mainly used in your chemistry. Other one is organic catalyst, students. Okay, here let's say example is platinum and everything. Now, organic catalysts are called as something called as enzymes. Examples, trypsin and all that, students. Trypsin, all that is an example of your organic catalyst called as enzymes. Now, with that being said, students, shall we start enzymes right now, students? Shall we start enzymes? So, enzymes are what, students? Enzymes are technically biocatalysts. Enzymes are biocatalysts. Now, what is the enzyme made up of, students? What are enzymes made up of? all enzymes are made up of proteins, except some of them have something called as a nucleic acid. The example of a nucleic acid, that is RNA, right? RNA enzymes. RNA will act like an enzyme, RNA enzyme, and that RNA enzyme is called as ribozyme. That is called as the ribozyme, students. So except the nucleic acid one, that is your RNA enzyme, where RNA can act as an enzyme, that is called as ribozyme. Ribozyme, students, ribozyme. So ribozyme is made up of your RNA enzyme, nucleic acid, but remaining all enzymes are made up of what? Proteins itself. Remaining all of them are made up of proteins, protein itself. Okay, protein itself. Okay, students. Now, some of you will not know what is an active site right now. So I'm going to make it very, very easy for you right now, very easy for all of you. So please understand what is an active site, because once you understand what is an active site, right, the next concept becomes so easy. Next is very, very important. The next topic, students, are very, very important. Next topics. Okay. Now students, active site, understand your active site. Okay. Now tell me, students, which type of protein can have 3D structure? Primary, secondary, tertiary, or quaternary? Generally speaking, the tertiary onwards we can see quaternary structure. Right? So we can see the 3D structure. So let's say we take a tertiary protein here. Our tertiary protein is 3D, like a woolen ball. 3D structure here, like a woolen ball. This is a tertiary structure right now of protein, basically, which is an enzyme in this case. Enzyme, students. Now in this protein structure, right, it has multiple foldings and everything is there. Okay. Now in this protein structure, there can be certain gaps or crevices or pocket. Students, in this protein structure of your enzyme, protein structure of this enzyme, right, enzyme is a protein. In this tertiary structure, there can be small gaps, or it can be called as your pocket or crevices, or we can also call it as crevices. Crevices basically means a crack. Crevices means a crack. This crack here has a functional group, and these small gaps or pockets or cracks or crevices found in the tertiary structure of protein, they are called as active site. Active site. Now I'll give you a better perspective. So from now onwards, please understand. Let's say here I have a lock. So how do you draw a lock? I've always forgotten how to draw a lock. Okay, let's say this is a lock here. How do you draw a lock? One second, students. How do you draw a lock? Students, I've forgotten how a lock looks like also. One second, students. 1 second. How do you draw a lock? Huh. Let's take this is a lock right now. Okay. Now, students, this entire lock represents this entire lock here represents enzyme. It represents enzyme, students. Okay, it's called as enzyme. All of you, voice aa raha hai na? Okay, now students, this lock is equal to the enzyme here, and this lock hole is equal to active site. So how we have on a lock can have something called as lock hole, or keyhole we say generally, but we say keyhole only, right? Keyhole, right? This keyhole is basically active site. So on this enzyme, we can have a active site, and this is a gap, right? It's a gap here. It's a gap here. Okay, students, it's a gap right now, students. It's a gap right now. Okay. Now, students, now let's say I have a key here. I have a key, students, a key. Now this key will go inside a lock here. Now this key is actually substrate. Key is substrate, students. So please remember always, students, please remember always, a substrate will always go join with the enzyme, not other way around. So technically speaking, in the entire reaction from now onwards, right, it is a substrate which will go and bind. Enzyme will not go and bind the substrate. No, it is a substrate which finds the keyhole, that is the active site. So this substrate will bind to the active site on the enzyme and the reaction will happen. Students, is that point clear to all of you? Students, is that point clear to all of you? All of you in the chat? All of you? Yes? Is that point clear to all of you, students? It is always the substrate which will go and bind to the enzyme onto the active site. Onto the active site, this substrate will go and bind. That is the main concept. That is the main concept for your lock and key mechanism. Okay. Now, students, you can see here also. This is a enzyme. This right here is a enzyme. Now this enzyme has, students, this enzyme has a small cavity, small crevices, small pocket or a small gap. That is called as your active site. To this active site, a substrate will come in bind. Always into this active site, this substrate will come in bind. That is the main concept here. Okay, students. Now let's understand why do we need enzymes? Like, what is the power of enzymes? Let's understand what is the power of enzyme. Okay. Now, students, here I have a normal reaction here, students. Normal reaction where CO2 plus H2O will give you carbonic acid, students. Carbonic acid. Now, understand, students. Understand. Now, this reaction without catalyst. Let's say there is no catalyst at all. In this case, let's say we will get 200 molecules of H2CO3 in 1 hour. In 1 hour without catalyst. Now, once you add a catalyst, that is carbonic anhydrase, in this case, catalyst. Now, once you add catalyst, students, you will get 6 lakh molecules of H2CO3 in, students, how much do you think it is, students? How much do you think it is? All of you. How do you think it can happen? 6 lakh molecules in 1 hour, 2 hours. See, here 200 molecules in 1 hour. So 6 lakh molecules in 1 hour. No. 6 lakh molecules, let's say in 10 minutes, 5 minutes, 1 minute, 30 seconds, 1 second. Students, in 1 second, in every single second you'll get 6 lakh molecules of H2CO3. 6 lakh molecule. 6 lakh molecule every single second in the case of with catalyst. That is the power of catalyst. That is the power of catalyst. All of you understood the power of catalyst? Crazy, you know, students, crazy, you know. So here we got 200 molecules in 1 hour, but right now with catalyst we are getting 6 lakh molecule in H2CO3 in 1 second. That is the power of your magicians, called as your catalyst, students, called as catalyst. Okay, catalyst, students. Now, students, I want all of you to understand there is this entire reaction. There is something called as a physical reaction, chemical reaction, right? Physical reaction, chemical reaction. That is a reaction happening. In physical reaction, there is no bond breaking. In chemical reaction, there is bonds are broken, bonds are formed. Okay. Two types of reaction. Now all of these reactions, there is a rate, how fast they can work. Okay, how fast they can work, that is right. So in an inorganic chemical reaction, similarly, the rate of physical or chemical reaction is referred to as amount of product formed per unit time. How fast are we getting the product in how much of time? That is, amount of product of formed per unit time is called as your rate of a reaction. Rate of reaction is amount of product formed per unit time is called as rate of reaction. Now if you give it a direction, that becomes velocity. Rate can be called as velocity if your direction is given. This is physics. I will not go into much physics part here. I will not go into much. Just remember that if you, students, tell me, all of you, very simple, I'll ask you, very simple. Let's say you're trying to make one sambar. If you keep the gas very high, will the sambar become faster or slower? Tell me. You're trying to make sambar right now. If you make the gas stove very hot, sambar will become faster. Sambar will become faster. If you keep the heat very low, sambar will become very slow. Same thing also, students. Same thing also here also. If for every 10 degrees change in either direction, catalyst reaction proceed at a vastly different higher rate than unsubtilized reactions. That is, if you increase the temperature, reaction can happen 10 liters generally faster. Decrease the rate or temperature, little slower it becomes. Very small, students. Rate of reaction can be changed. Okay, but this is again contradictory because lot of enzymes cannot work at different temperatures. So this is a little contradictory here. Now, students, now all of you want to understand, students. I want all of you to understand what actually is happening. Like all of you understood enzymes are amazing right now. All of you know enzymes are amazing. But how are they so amazing? Like, what is so cool about enzymes? What are they actually doing it to make it so amazing? Like, what are they even achieving? How do they even do it? Students, do you want to know? Like, how are they able to make 6 lakh molecules in 1 second? How? Students, all of you want to know? Students, do all of you want to know? Like, how is it even possible? How is it even possible to get 6 lakh molecules? What is so special about them? Like, what are they even changing there? Students, do you want to know? All of you. So do you want to know? All of you want to know or not? Want to know? No. How are they doing it? What is the secret? Let's follow. Let's understand. Okay. Let's understand the secret, students. Let's understand the secret right now. Okay. Now I want all of you to understand, students. I want all of you to understand there is something called as activation energy. Before you solve the mystery behind it, let's understand something called as activation energy. Very simple concept, understand, students. Now let's take, I have a substrate. This substrate will now turn into the product. Now this substrate will turn into product. Now when this conversion is happening, generally right in the middle, students, right in the middle, this stage here, this stage is called as what? It is called as transient phenomenon. Okay, transient phenomenon. In this transient phenomenon, there is something called as transition state is found. So between your enzyme to product, this conversion is happening, right? There is always in the middle, there is a very unstable, and high energy intermediate is found. High energy intermediate is found here, right in the middle. Right in the middle, we have some very high energy transition state. That is, this substrate will change into enzyme substrate complex. This enzyme substrate complex eventually turn into product plus enzyme. So this substrate doesn't become product generally. Right in the middle, there is a transition state. And this, students, this transition state has very high energy, very high energy, very high energy. So students, what's the definition then? Definition of activation energy, students. Definition of activation energy is that it is the, if you can see students, this is your substrate, this is the product here, right in the middle we have the transition state. This is your transition state here, transition state. Okay, this transition state will have very high energy and very unstable here, very unstable. Now students, your initial substrate and transition state, is there a difference in that? Transition basically means intermediate or something in the middle. Transition means something in the middle. Okay, now this substrate and transition state, do they have difference in energy? All of you, if you see this one also, this is substrate here, all of you, substrate, potential energy of the substrate is somewhere here and transition state is right here on the top. So students, right in this gap here, do you see a energy difference, students? All of you students, do you see energy difference here? Yes or no? There's a difference in energy. Yes. Now the difference is called as activation energy. All of you students, it is the definition wise. If you go, so what is the definition? Definition is very simple. Definition is that activation energy is nothing but, it is the average energy difference between the substrate and the transition state. So some energy this has, some energy this has. Now whatever is the difference, middle part here, this difference here, that is called as your what, students? That is called as what? Activation energy. See students, I would have given you this definition directly, but if I give you the definition, you will not understand, right? So if you can see here, this person has some energy, this person has very high energy. So difference between that energy is called as what, students? Charmander has some energy, Charmeleon has some energy. Charmeleon has very high energy, this person has very low energy. The difference in the energy is called as activation energy. Now students, I need a volunteer. Students, I need a volunteer now. All of you need a volunteer. Who wants to volunteer? I need a volunteer in the chat. Students, I need a volunteer. Who will come? Students, quickly, I need a volunteer in my class. Students, you know, I always teach with all of you. You are my test subjects. All of you wants to volunteer, students. Who wants to volunteer in my class? Who wants to volunteer? Okay. Now let's take Sharmita on, students. Sharmita is very regular student right now, part of our DNA also, right? Let's take Sharmita right now. Okay, students. Let's say this is Sharmita here, students. Which color hair? Students, which color hair? We'll give pink color hair to her. We'll give pink color hair to Sharmita. Okay. Now, students. Now, this Sharmita right now is trying to do something called as high jump. Okay, students. All of you, Sharmita is trying to do high jump right now. Okay. Now, if she is trying to jump over this and she is trying to climb this wall. Let's say she's trying to climb this wall. In this wall, there are steps here. Okay. Let's say there are around 50 steps. Okay. And this side, she has to come. Students, how long do you think she will take right now, students? Tell me, how long do you think she will take? Let's say Sharmita will take to climb the 50 steps and go this side. Let's say she will take around 30 minutes time. Now, this is one condition. This is one condition. Okay, this is one condition right now. Okay. Now, in the second condition, what is happening, students? In second condition, what is happening? Now, This is magenta actually. In this case, next day, this is day 1. Let's say this is day 1. She has learned her lesson now. Now, Sharmita in day 2, what will happen? She has to climb the stairs. Now she'll call a friend. Bro, please, I have a problem. I cannot climb the 50 stairs. I need help. A friend will come. He's like, dun, dun, dun. The friend will come now. The friend will come and he'll completely break. Talk, 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 talk. Everything, full break. Breakage will happen. Okay. And this wall will become this much. This much. Okay. Now here we have hardly 1-2 steps. Now students, tell me, how long do you think she'll take right now? How long do you think she'll take? She will take, students, how much? She'll take, let's say, 30 seconds. 30 seconds right now. 30 seconds. Students, do you realize the difference here? This entire wall, Sharmita is the substrate. Substrate here. This large gap, this you see, right? This is called as activation energy. Here we have, this is called as your transition state. Substrate, here we have product. If Sharmita has to become, this substrate has to become product, it has to reach the energy. The activation energy has to be met to reach transition state and then become here. Now students, who is the friend here? The friend here who came and helped is called as enzyme. Enzyme is the friend here. Now students, what did enzyme do? Enzyme came here and it completely reduced. Reduce what, students? What is reduced here? Tell me students. Tell me students, what is reduced here? This entire activation energy. Entire activation energy is reduced by the enzyme. So this entire activation energy is, this is now transition state now. This is now the transition state here. Okay. Now this entire activation energy is reduced now. So substrate can become product easily. From 30 minutes it became 30 seconds. From 30 minutes it became 30 seconds right now. Why? Because enzyme came into picture. Enzyme decreased the activation energy. That is how enzymes work. Enzymes will always work in a fashion that there is an energy difference, they'll reduce the difference only. Simple as that. Students, how many of you understood? Students, how many of you understood? See here, substrate, this is activation energy without enzyme, very high energy. Students, can you see? Very high energy is required. Now, as soon as you add enzyme to it, the activation energy is reducing here. So, substrate, this is the activation energy with enzyme. Easily it became product right now, students. Easily it can become product right now. Okay. See students, here also. Without enzyme, very high activation energy. With enzyme, very low activation energy. Simple as that, students. Simple as that. Simple as that, students. All of you understood? Students, all of you understood or not? The simple concept of activation energy. All of you. Students, see here. See here. Reactant, transition state, activation energy, product here. Very high activation energy. Takes a lot of time. Now you add a catalyst. This thing gets reduced right now. Reduced. Activation energy for the catalyst pathway is less than the uncatalyzed pathway. So reactant can easily become the product now. Easily become. So uncatalyzed pathway, catalyzed pathway. Simple as it gets, students. Simple as it gets. Clear, all of you? Clear? Is the concept clear to all of you? Yes or no, students? Yes or no? All of you understood? Students, all of you understood? Okay. No, students. No. Now, what is the mechanism? What is the mechanism here? Students, mechanism is very, very simple actually. Mechanism is very, very simple. Okay. Mechanism is, what is the mechanism here, students? Initially, we have the substrate. This substrate forms a bond with the enzyme. So we get enzyme substrate complex. Now this becomes enzyme product complex. This now becomes what? Substrate becomes substrate becomes product. So we have enzyme product complex. Now it becomes product is separate here and enzyme is separate here. So substrate becomes product here. First, enzyme will form a bond with substrate will form a bond with the enzyme. Then we have your enzyme product complex, enzyme substrate complex, then separate out. Students, very simple mechanism. Students, very simple mechanism. Done, students? All of you understood? All of you? Done? Substrate, product here. These are the middle intermediates right now. So as you can see, we have enzyme, active site, substrate. So this will come and join here. We get enzyme substrate complex. Eventually, the product is separate. This entire thing both gets mixed up. Product, enzyme is separate. This is what happens. This is what happens in the case of your reaction. Now, students, now all of you, there is something called endothermic reaction, exothermic reaction, students. Endothermic and exothermic reaction. What is the difference? Difference is very simple, students. Difference is very simple. Endothermic versus exothermic reaction. Okay. All of you, see this. In your endothermic reaction, can you see reactants here, students? Here, reactants have less energy. So endothermic will happen what? In endothermic, these reactants will absorb energy, students. They'll absorb energy. So endothermic reactant has low energy, they'll absorb energy and they'll become product here. Endothermic reaction to reach the activation energy. Now in exothermic, reactants are very high energy always. So in exothermic, heat is released, students. Heat is released. So when the heat is released, we get the product easily. When the heat is released. So in endothermic, reactants are less energy. Exothermic reactants are more energy. I hope this is clear to all of you, students. Endothermic versus exothermic. Endothermic versus exothermic reactions. Okay. Now here is a simple what exactly happens in the entire process here. First, the substrate will bind to the active site of the enzyme, fitting into the active site. So substrate is coming and binding here. The key is coming and going into the lock keyhole. Binding of the substrate induces the enzyme to alter the shape, fitting more tightly around the substrate. That is, after the lock has gone inside, after the key has gone inside, the lock will change the shape and strongly it will hold. The active site of the enzyme now is close proximity to that of the substrate, breaking the chemical bonds of the substrate, and new enzyme product formation is complex form. So we get enzyme product complex we get, and later on they are released eventually. Students. Okay. Now students, now there are something called as factors affecting enzyme activity, students. Factors affecting the enzyme activity. Okay. There is temperature, pH, substrate, enzyme concentration, inhibitors. Now, if you're done with this, chapter is almost over. Right now, students. Okay. Now, what about in the case of temperature, students? Temperature. Students, I want all of you to understand every single enzyme is special. They are very uncooperative, very rigid they are. That is, if you see, enzymes are basically what, students? Enzymes are basically proteins. Yes or no? Enzymes are basically proteins. Now, if this proteins temperature goes very high, what happens? In this temperature goes very high, protein will denature. If temperature goes very low, protein becomes inactive. So, high temperature is a risk, low temperature is a risk. So, all of these require very proper temperature, very optimum temperature. And students, generally speaking, enzymes generally work at very low temperature only, very medium temperature. Because try to imagine, right? If all of these proteins, they cannot survive at high temperatures, like your inorganic catalyst, which can work at very high temperature, platinum and everything, your enzymes cannot work at high temperature. But there is an exception, right? There is something called as thermus aquaticus. Thermus aquaticus is obtained from hot springs. So thermus aquaticus can give an enzyme called as Taq polymerase. Students, you might have heard it before, right? In the which chapter, students? Biotech chapter, PCR, you might have heard in PCR, right? Something called as Taq polymerase. Now this Taq polymerase enzyme can function very well even at high temperature. Why? Because the Taq polymerase enzyme is obtained from a organism called as Thermus aquaticus. This Thermus aquaticus can function it is surviving in hot temperature. So there are exceptions. Okay, there's exceptions are there. No, students, no. See, high temperature denature, low temperature inactive. Very simple, students. Same goes for students, same goes for pH also. There is something called as optimum temperature, optimum pH. Anything goes here and there, they will not work. They're like, I will not work, students. You can see here. Anything goes here and there, they will not work only. See here. Can you see? Pepsin at this, salarium at this pH, aluminum this pH. So apart from the actual proper pH, if there is something here and there, they'll not work at all. They'll not work. They're like, why should I work? They're like, why should I work at all? Okay, so there is up to optimum pH. Now, what is the meaning of, how do you think substrate concentration plays a role, students? How do you think substrate concentration can play a role here? Okay, now for this, I want all of you to tell me, what is your favorite food item? I'll teach you with the help of a simple example. Tell me, students, what is your favorite food item? Favorite food item, all of you. Tell me, students, what is your favorite food item? All of you. Yes? Okay, let's take dosa only. Dosa or dosa. Okay, students, let's take, all of you love dosa, yes or no? All of you love dosa? Yes or no, students? All of you love dosa? Even I love dosa, students. I love dosa. Okay, now one small scenario, students. One small scenario. I will call all of you to my house. Okay, all of you coming to my house for a function. Now all of you sitting in my house, let's say I give on the table, I make all of you sit. Now all of you are right now what? All of you right now are enzymes. All of you are right now what? Enzymes. Okay. Now students, this dosa is the substrate. Now all of you are hungry. Yes or no, students? All of you came to my house. All of you are hungry now. You are the enzyme. You are very, very hungry here. You are the enzymes. You are the enzymes. You are very, very hungry right now. You come to my house. Now students, I will start giving you dosa right now. I will start giving all of you dosa. I'll give you one dosa, second dosa. Yes or no? So as the time goes by, your stomach will become full. Yes or no, students? Yes or no? Your stomach can become full. Let's say some of your stomach can become full at 3 dosa. Some of your stomach can become full at 7 dosa. Some of you can eat 10 dosa. Okay, very simple rule. So here also same thing. These are the enzymes. Now the rate of reaction will depend on, right? The rate of reaction will keep on increasing. That is, your stomach will become full and full and full. Your stomach will become full and full. The more I increase the substrate, this is called as reaction rate here. Here is your substrate concentration. Okay. Now, the more I increase the substrate, the reaction has become faster and faster. So let's say here you are, I'll give you substrate, you will start eating the substrate. Now after some time, can you see here? This person's stomach is full, 2 dosa full. This person 3 dosa full. Some of you still hungry. Okay. Now let's say after 10 dosas, right now, that is substrate concentration. Substrate concentration is very high now. I've given 10 dosa. So at the end of 10 dosas, can you see every single enzyme is filled right now? It is filled. So all your stomach is filled right now. Now students, tell me in the chat right now, if I start giving you more dosas after this, will there be a change in your stomach? Is there any space? No. Your hunger is already satisfied. It cannot go any further. You will end up vomiting. Right? That is very bad. So at one point, how much ever dosa I give you, you cannot eat. You cannot eat after that at all. Right? Similarly, at one concentration where all the active sites are filled here, how much ever substrate I increase, Will there be a change in reaction? No. Students, what is the takeaway right now? Takeaway is very simple. Takeaway is that with the increase in substrate, rate of reaction will also increase. You increase the substrate, rate of reaction will increase, but only up to a certain limit. That is, at a point we reach something called as saturation point. After saturation point, how much ever you add, no, nothing will happen, students. Nothing can happen after that. Nothing can happen after that. Okay. Now that saturation point is where you're full. Your stomach is full right now. You cannot increase your food. Okay. Saturation point is reached right now. Okay. So this is called as what? How substrate concentration affects the rate of reaction. Here we learnt how pH interacts, how temperature interacts. That is temperature acting to affecting the rate of enzyme activity. Right? Enzyme activity. This is the substrate concentration. So initially it will increase, but after some time stomach is full. After some time the reaction cannot increase anymore. Now students, now I want to know at what velocity, right? There's something called as Vmax here, students. Something called as Vmax. Vmax is nothing, students. Vmax is nothing but maximum velocity at which all active sites are filled. That is, how fast did your stomach become full? Student, understanding, how fast do you think your stomach became full? Like, it is very difficult, right? Sometimes you can't tell. Was it 3 dosa? Was it 4 dosa? Was it 7 dosa? You can't tell. Very difficult. So instead of seeing at Vmax, we look at half Vmax. We look at velocity at which half of active sites are filled. Like, I can't know, I cannot tell, students, I cannot tell when do you reach saturation. I can't tell. Like 10 dosa, 8 dosa, but I can assume that somewhere in the 4 dosa you became some full, yes or no? In 4 dosa you became full. That is called as half Vmax. That is velocity at which half of the active sites are full. That is, let's say someone from your chat, somewhere here, you can see, students, somewhere here we reached half Vmax. If this is Vmax, maximum velocity at which everything is filled, somewhere here we have half Vmax, where half of the active sites are filled, where half stomach is full. Now the question arises, how many dosas did it take to fill your stomach half? Tell me, students, tell me, all of you, how many dosa, students, how many dosa did it take to fill your stomach half? How many? Anyone? How many dosa did it take to fill your stomach half? That is called as Km. That is called as Km value, Michaelis constant. Okay, that is called as your Km. That is nothing but students, concentration of dosa, that is substrate, at half Vmax. That is, when your stomach, students, when your stomach was half full, no, when your stomach was half full, how many dosa did it take, students? How many dosa did it take? That is called as Km value. That is concentration of substrate at half Vmax. So if this is half Vmax, in this half Vmax, what was the substrate level? That is called as Km value, Michaelis constant. Students, how easy is it to understand? Tell me now. How easy it is to understand to what is Vmax, half Vmax, Km value? Basically, Vmax is how much time did it take? How fast did your stomach become full? Half Vmax is when did your stomach become half full? And when your stomach was half full, how many dosa did you eat? That becomes Km value. Students, that becomes Km value, students. How many of you understood? Raise your hands. Students, how many of you understood? Raise your hands quickly, students. Raise your hands. This is your stomach is half full here. So that means substrate concentration. Students, I'll teach you one more small concept here. Please understand. Let's say here are two reactions happening here. This is called as Vmax. This is called as Vmax. This is half Vmax. Somewhere here is half Vmax. Tell me, students. Now, this half Vmax, this is the Km value. This is the Km value here. Okay, tell me, students. Now, students, let me ask all of you one very simple question. How can I profit? Can I get a profit if I make you eat 5 dosas? Can I profit if I make you eat 2 dosas? Tell me, when will I get profit? When will I get profit, students? When will I get profit? All of you. When can I get profit? When I'll make you eat only 2 dosa, right? Yes or no? That's when I can get profit. If I make you eat 5 dosas, loss for me only, no? Loss for me only. Students, understand very important concept. A good enzyme will have less Km value. So if this Km value is here, this Km value is right here, this is a good enzyme because less substrate is required to reach half Vmax. I can fill your stomachs in 2 dosa only. Good. That is a good enzyme. That is a good enzyme. Here, this is a very okay enzyme. Very okay enzyme because it is taking more substrate. It is taking more substrate to reach half Vmax. So it is okay substrate. This is a very important concept. That is, good enzyme will always have less Km value. Less Km value because it requires less substrate to reach half Vmax. Your stomach is half full. With 2 dosa only. Good for me only. So, okay. Students, how many of you understood this concept? Dosa theory. Okay, students. Now, students, I'll teach you what is inhibitors. Let's start inhibitors, students. Inhibitors, very simple. I will show you animations and I'll teach you inhibitors. Animation time. Okay. Now, students, what are inhibitors? First of all, students, what are inhibitors? Okay. Inhibitors are chemicals that shut off enzyme activity. They're like, they will stop the enzyme activity completely. They're like, enzyme, no entry. No entry for the enzyme here. No entry for the enzyme here. Okay. So, chemicals that shut down the enzyme activity. In other words, inhibitors can inhibit the enzyme activity. Okay. So, if this is your substrate, it has to go and bind here. Inhibitor will come and bind somewhere here and it will stop the reaction. It will completely stop the reaction only. That is called the inhibitor, student. That is called as inhibitor. Now, inhibitor, there are 3 types. Competitive, non-competitive, uncompetitive. Difference you understand, student. Difference you understand. Now, first of all, students, what is competitive? Understand, students. What is competitive? All of you. Understand what is competitive inhibitors? Competitive. Okay. See, students, here. This will come and bind. So, this and this, will both them look alike here? All of you. Do they look alike? This is your substrate. This is your inhibitor. So, in the case of competitive inhibitor, the competitive inhibitor what? It will resemble the substrate compared with the substrate of the same active site. So, instead of your substrate going and joining, inhibitor will take the place. Students, instead of substrate going and joining, inhibitor will go and join. Inhibitor is like, I'll go and join now. Okay, that is called as competitive. It will do competition now. Competition. So, inhibitor will go and join. That is called as inhibitor complex, competitive inhibitor. Okay. Now, students, now, what is non-competitive, students? Non-competitive. Now, non-competitive students will not join from the front. This person will wait now. Non-competitive inhibitor will not compete from the front. It will go from back side here. Can you see? It will go from back side and it will change the shape here. Change the shape. So, your substrate cannot bind now. See this. Binds at the site other than the active site. Change the shape of the active site such that substrate is not able to bind. It can bind to free enzyme or enzyme substrate complex. It doesn't matter. So, it will come from behind, bind off, change the shape, This cannot bind anymore. Our substrate cannot bind anymore, students. So we get enzyme inhibitor complex, non-competitive. It will not do competition only. It will come from backdoor. Okay. Now what is uncompetitive? Uncompetitive. Uncompetitive is the worst kind. Why? Because students, why? Because uncompetitive will always wait. See students. Uncompetitive, this uncompetitive enzyme. It will inhibit, will not join here. It will not join initially. It will wait. Once this joins, then it will join, kick it out. Okay. It will bind to the enzyme at site other than the active site, yes, but it cannot bind to the free enzyme. Free enzyme it cannot bind, so it will wait. Once it binds, bind here is removed. That is called as uncompetitive. Students, that is called as uncompetitive, students. Uncompetitive. This person will wait always. Once it binds, bind from behind, kick it out. The worst kind. Uncompetitive is the worst kind, uncompetitive, students. Worst kind of all. Okay, so students, in your NCRT you only have competitive, so learn only competitive, students. Okay, example of competitive is your malonate. Now this is an enzyme, this is succinate, right? This is your substrate here. Okay, this substrate will go and bind to the enzyme. This substrate will go and bind to the enzyme like this. It will go and bind, okay, like this, and we get fumarate eventually. Now what happens, students? This malonate is there, right? Malonate. Doesn't it look like your succinate only? Tell me, students, doesn't malonate, see, structure is CCO, CH2, almost looks alike here, this part looks alike. The only difference is that here we have an extra CH2, here that extra CH2 is missing. So it looks alike here. So this malonate will go and bind with the enzyme now. See, malonate enzyme complex and no product here because your inhibitor is going and binding with the enzyme. And this inhibitor will look like your substrate. Copycat it is doing, copycat. Okay, now students, that is your entire thing. Temperature, everything, students. Students, please remember, all these factors will affect the protein structure of your enzyme. And enzymes are generally in your tertiary structure. Like see here, the activity of the enzyme can be affected by a change in condition, which can alter the tertiary structure of protein. So tertiary structure of protein can be affected here. It's a very important line. Okay, very important line, students. You can see, students, here Vmax, half Vmax, substrate concentration is Km value here. Okay, so here is a question like that. Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of succinate dehydrogenase. Okay, it is succinate dehydrogenase because it will look like succinate dehydrogenase. Stop the activity, students. Stop the activity completely. Okay, now students, this you can study on your own. It's very, very simple. Here there was a NEET PYQ here. They have given direct definitions here. This you can read, students. Very easy it is. What is oxidoreductase? What happens here, students? We have one reductase, one oxidase with redox reaction. Transferase, what happens in transferase? S plus G, GS, S is there. S will go and join, G will be removed out. This is basically like your displacement reaction. Okay. Lyase, hydrolase. Okay. Isomerase, all of you know. If you use it in your glycolysis also. And ligase is joining. This you can study. This you can study on your own. Okay. Now what I'll teach you, the last part I'll teach you is the type of enzymes. Okay. Now there is something called a simple enzyme which only has your protein part. Then there is conjugate enzymes. Conjugate enzymes has a protein part and a non-protein part. Understand, students? Understand here. So conjugate enzymes, simple enzyme will only have protein part. Only protein is there. Enzyme is made up of only protein. But conjugate enzymes are made up of protein and non-protein. Both parts are there. Okay. So it has a protein part is called as your apoenzyme, which is biologically generally inactive in nature. This is cofactor. Cofactor is non-protein part. So when this cofactor, it will join with the apoenzyme. This becomes activated and it is called as holoenzyme that time. See here, students. So when this apoenzyme protein part, cofactor is your non-protein part, right? When they join together, when we take the protein part and non-protein part, join together, they are called as holoenzyme. They are called as holoenzyme right now. Holoenzyme. And this holoenzyme is biologically active. Only apoenzyme, inactive, not possible. So once you add the cofactor, it becomes biologically active. It becomes biologically active that time. Biologically active. Okay. Now students, here I'll just, okay. Students, Kavita, I only teach mostly zoology. So I teach botany mainly, so you have to follow Gopika ma'am also. You have to follow Gopika ma'am also, students. Gopika ma'am also. One second, students. One second. No, students. Okay. Now, students, you can see here, these are different types of cofactors. Here I told you, students, right? Cofactors are non-protein part. There are mainly three different types of cofactors. One is called as prosthetic group, one is called as coenzyme, one is called as metal ion. Now, what is the difference, students? Prosthetic group is always organic, coenzyme is organic, while metal ion is inorganic in nature. Prosthetic group is very tightly bound to the apoenzyme, very tightly bound to the protein part apoenzyme, very loosely bound here, and it will show coordination bond here. Now, what in examination they'll ask you is examples. They'll ask you directly examples. Tell me the example of prosthetic, or zinc is an example of, that is your metal ion. Okay, so to understand here, hydrogen peroxide gets converted to water plus oxygen. Now here, which is the enzyme? The enzyme or the protein part is called as peroxidase. Now, if there is only peroxidase, it is incomplete. It cannot work at all. This protein part need a non-protein part also, cofactor, right? The cofactor here is heme group. Heme group is the cofactor here, students, here. So without heme group, it cannot work. The partnership cannot work. The heme group, students. The heme group is the prosthetic group, cofactor, heme group. Similarly, in the case of coenzyme, they've given you two examples. NADH and NADPH, nicotine amide adenine dinucleotide. Those are the two different types of your coenzymes in a lot of reactions. And these two coenzymes, they are made up of vitamins. They're made up of vitamins. One example is given is niacin. One example is given is vitamin niacin. So NADH and NADPH has niacin in it. They have niacin. Now metal ion, students. Can you see here? Carboxypeptidase is an enzyme. Carboxypeptidase is an enzyme. This enzyme is a protein part. It has a non-protein part also. That is zinc. Zinc is the cofactor here, students. Zinc is the cofactor. That is your, all of these are called as your cofactors. They are non-protein part which help in the chemical reaction. Okay. So you can read the NCERT lines. It's very important here. Non-protein part is called as the cofactor. Protein part is called as your apoenzyme. Yes or no? The protein portion is called as the apoenzyme here. Everything is given, students. Coenzymes, metal ions, everything is given, students. And with that, right, niacin is a type of vitamin niacin, vitamin. Okay, that vitamin is part of your coenzyme there. Okay, so with that, chapter is over. With that, all of your chapter is over, students. Now I have a very, very simple question to all of you. Very, very simple question, which I want to see in the comment section. All of you, very simple comment section, students. Very simple question. Okay, the question is very, very simple. Tell me, students, what is the monomer of inulin? What is inulin made up of? Very simple question, right? Very simple question. What is inulin made up of? Inulin is a polysaccharide. What is it made up of? What is the monomer in inulin? That is a simple question to all of you. Students, I will see in the next class. Next class, I'll be starting with plant kingdom. I'll be starting with plant kingdom for all of you. We will go plant kingdom in 4 parts, right? 4 parts, right? Or 4 parts. We'll do 4 parts of plant kingdom. Okay, students. So answer in the comment section, not here. Answer in the comment section. Along with that, in the comment section, tell me how was the class, and also did you enjoy the class? Did you enjoy the dosa theory? Because with the help of dosa theory, right, all of you understood what is Vmax, maximum velocity at stomach is full, half Vmax, maximum velocity of half stomach is full, Km value. When your stomach is full at half Vmax, how many dosa you ate? That is Km value. Yes or no, students? Yes or no? Right. So it's very easy. Biology is very, very easy. And you will realize, students, every single time I teach a chapter, the chapter becomes very easy for you to understand. Okay. So thank you so much, students. Baba, take care all of you, and I'll see you in the next class. Until then, keep studying, keep the grind on. Okay, students. Students, if you are not part of DNA batch, you can ask other students here. Other students here who are already part of DNA batch, right? Ask them once how amazing DNA batch is, how I teach morphology. I am doing morphology right now there. How was the morphology class? Just ask them once and they'll let you know how was DNA batch morphology. Killer visualization, killer it was. Okay, students. Thank you so much, students. Bye bye, all of you. Take care and have a great day ahead, all of you. Thank you.